Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's weekly seminar series. My name is Paul Jakes and I'm a senior scientist at the HNRCA and host of today's seminar. Before introducing today's speaker, I have just one minor housekeeping announcement. As always, we welcome your questions for our speaker, but I would like to remind you to ask your questions using the Q&A icon located on the webinar toolbar, not the chat box. And I encourage you to add questions as they arise during the presentation. Let me now welcome today's speaker, Dr. David Baer. Dr. Baer is a supervisory research physiologist with the USDA Beltsville Human Nutrition Research Center. He serves as the research leader for the center's Food Components and Health Laboratory. Dr. Baer's research focuses on controlled dietary intervention studies that investigate the relationship between diet and risk for chronic degenerative diseases, especially cardiovascular disease, cancer, and diabetes. In those studies, he has investigated the role of dietary patterns, individual foods and food components, and food processing. The foods and food components that he has examined include, among others, different types of protein, fats, fiber, margarine, butter, plant sterols, nuts, whole grains, berries, alcohol, and tea. In addition to dietary intervention studies, Dr. Baer is involved in research studies to validate food survey methodologies and to develop new methods for dietary assessment. Today, Dr. Baer will discuss some of his work on foods, food processing, and dietary patterns in a presentation entitled "What Meat to Eat: Insights on Food Processing and Dietary Patterns." And with that, let me turn the microphone over to Dr. Baer.
Thank you, Paul, for the kind introduction and the invitation to share some results from a recently completed dietary intervention. As you just heard, that's been the focus of our lab's research program for many years. What parts of the diet, food components, traditional nutrients, bioactives, and those sorts of things affect risk for a variety of chronic diseases. What I'm going to share with you today are some results, some early results from a dietary intervention study that we finished last year, and we've been doing analysis of samples and statistical analysis, and we have some data to share today. It's the first time these data are being presented, and this was a study that focused on meat and meat processing and different dietary patterns. Let me start by acknowledging both USDA and the Beef Checkoff, who helped fund this research, and a lot of people who were involved in making this research come together. First, I want to acknowledge Janet Novotny, co-principal investigator on this project, a couple of our statisticians, Matt Kramer and Nora Bello, and Rita Rawal, who is a postdoc fellow and serves as our clinical research coordinator and really helps bring all the bits and pieces of this work together. And then on the left-hand slide, and the list of names of the team in the Food Components and Health Lab who work on sample collection, sample analysis, and everything required for the intervention. And all the people listed on the right from our metabolic kitchen under the leadership of Patrick Sullivan, who also makes sure that everything gets done in a timely manner. So you can see from this list of people, it takes a lot of hands to get this done. And we're very appreciative of everybody's efforts. So let me start with a couple of definitions about meat. We're mostly going to be talking today about meat and meat processing. Let me start with the Dietary Guidelines for Americans, the current version, which defines processed meat as meat, poultry, or seafood products preserved by smoking, curing, or salting, or addition of chemical preservatives. It's a relatively short and succinct definition, but it has some problems in terms of really describing what's going on. There is also a meat science lexicon. This was a lexicon that was published in 2018 that brought together meat scientists to sort of describe in more detail about what really happens with meat. And their definition, this meat science lexicon, really divides meat into minimal processing and further processing, where minimal processing is any process where raw, uncooked meat products have not been significantly altered compositionally and contain no added ingredients, but may have been reduced in size by fabrication, mincing, grinding, and or a meat recovery system. So minimally processed meat, obviously all meat that we consume is processed. It's butchered to some degree, and it might be ground. That would be minimal processing. Further processing is any process where meat products undergo transformation beyond minimal processing that contain approved ingredients and may be subjected to a preservation or processing step or steps through which the application of salting, curing, fermentation, thermal processing, which could be smoking or cooking, batter or breading, or other processes to enhance sensory, quality, and safety attributes. So the meat science lexicon is a lot more involved, but also incorporates more descriptive terms of what happens once meat is butchered and available for further processing. So just one thing that the Dietary Guidelines for Americans doesn't capture is something like breading. So chicken nuggets would be considered further processed with the meat science lexicon, but not necessarily considered processed with the DGA definition. And we'll talk a little bit more about definitions because there's been so many different ways people have described meat. People have captured information about what people are eating, and that has really led to a lot of confusion. So just a brief history. There are historical records that suggest processed meats have been part of the human diet for 5,000 plus years. Processing does several things. It extends the shelf life to an otherwise perishable food. So salting has been around for centuries because it helps preserve food. Processing can reduce waste through the use of lesser quality cuts of meat, and also provides consumers with convenience, flavor, and cultural identity. The questions about meat and health have been around for a long time. Even in the 1950s, some scientists suggested that meat consumption, and at this point in time, they weren't really looking at processed versus unprocessed or minimally processed versus further processed. It was just some concerns about meat consumption and increasing risk for coronary heart disease and cancer. And even in the 1950s, some of these concerns resulted in policies and dietary recommendations to limit meat intake. With meat consumption, the main focus has been on cancer and heart disease, cardiovascular disease, including coronary heart disease and stroke. More recently, there's been some data coming out suggesting relationships between meat intake and risk for type 2 diabetes. Today, we're going to focus mostly on the cardiovascular disease aspects. And those will be the data I show will be things related to cardiovascular disease. Next few slides, we'll talk a little bit about dietary recommendations, meat's contribution to the overall diet, and some associations between red meat, poultry, processed meat consumption, and some health outcomes. So let's just take a look at some different dietary recommendations. The current dietary guidelines for Americans states that most intakes of meats and poultry should be from fresh, frozen, or canned, and in lean forms, like chicken breast or ground turkey, versus processed meats, like hot dogs, sausages, hams, luncheon meats. One of the interesting things about this recommendation is the canned. You can find canned meats in the marketplace, Most of these would be considered processed. All the ones that I have found are have at least salt added to during that canning process. But the focus here is on lean forms of meat. American Heart Association recommends choosing healthy sources of proteins, mostly from plant sources, regularly eating fish and seafood, substituting fat-free and low-fat dairy products in place of full-fat versions, and for people who eat meat or poultry, choosing those that are lean and unprocessed. So American Heart Association, their main focus is on low-fat diet, plant protein sources, fish and seafood consumption, and limiting fat intake. American Institute for Cancer Research, their recommendation is to eat no more than moderate amounts of red meat, such as beef, pork, and lamb, and eat little, if any, processed meat. Now, people eat meat for a variety of different reasons. These are some data from a paper published in 2019, focusing on omnivores, on people who eat meat, and asking the reasons why they consume meat. And in this cohort, the people who consume meat, their number one reason is that they consider meat an essential component of a healthy diet. They enjoy eating meat. They feel that meat is part of their traditions. And they believe they lack the knowledge and cooking skills to prepare an adequate meal without meat. So these are some of the key reasons why meat consumers choose to consume meat. Now, let's talk a little bit about how, in this case, beef consumption contributes to nutrient intakes. On the left-hand side of the slide are data from pregnant and lactating women, and on the right-hand slide are data from 60-plus-year-olds. These were data from two publications that were published earlier this year in Nutrients by Sanjeev Agarwal and Vic Fulgoni. So they were using NHANES data over several cycles and looked at the nutrient intakes of non-consumers versus consumers. So we see a general pattern here that both for pregnant and lactating women and for older individuals, their protein intake is higher for beef consumers versus non-consumers. Calcium intake is higher for beef consumers versus non-consumers. No effect on potassium. B12 intake is higher. And again, this isn't so much a concern for pregnant and lactating women, but certainly for older individuals. Vitamin B12 intake is higher for consumers of beef versus non-consumers. And iron intake is higher for consumers of beef versus non-consumers. The nutrients in purple, the calcium, potassium, and iron, those are nutrients of public health concern, iron for pregnant women, as described by the Dietary Guidelines for Americans. So we can see, and we would expect things like vitamin B12 and iron to be higher in consumers because beef is intrinsically contains relatively high amounts of B12 and iron. But one of the interesting things in these data is that both for the pregnant and lactating women and the older individuals, calcium consumption is higher. Calcium intakes are higher for consumers of beef versus non-consumers of beef. And this is kind of interesting because beef is not a particularly good source of calcium. It's actually quite low in calcium. So this must reflect some other component of the dietary pattern that is contributing to calcium intake. And in fact, the authors of the papers suggest that dairy intake might be associated with this higher calcium intake. So beef consumers, as part of their other aspects of their dietary pattern, might be consuming more dairy than non-consumers of beef, and therefore their calcium intakes are higher. So there are some key nutrients for which consumption of beef, and there's some other data in the literature on pork and poultry. Consumption of meat can contribute to intake, including intake of nutrients of public health concern. We see in red that sodium intake is higher in consumers of beef than non-consumers for both of these cohorts, the pregnant lactating women as well as the 60-year-olds. That is generally associated with a bad thing. And again, sodium is not intrinsically high in beef. So this also is probably reflective of other aspects of the dietary pattern. Now, there are a lot of meta-analyses and systematic reviews looking at meat consumption and risk for a variety of diseases. We're just going to look at a couple because we want to get to some data. This is a meta-analysis of randomized controlled trials focusing on cardiovascular disease risk factors. This was published in 2017. So the inclusion criteria for this meta-analysis had to be an RCT, aged 19 or older. The intervention group had to consume at least half a serving of total red meat per day compared with the control group. And there had to be at least one CVD risk factor reported. The meat, that half a gram or half a serving higher or lower, was all forms of beef, pork, lamb, veal, goat, non-game birds, and included preserved meat, further processed meat, including smoking, salt, curing, salting. So all meat. The comparators were all lumped together and could have been fish, either lean or fatty, seafood, meat supplement, chicken, tofu, plant protein, whole grains products. So the meat could have been any type of meat, and the comparator really was anything that wasn't red meat. And these studies included weight loss studies, people with heart healthy dietary patterns, and a variety of other dietary patterns. What they found in this meta-analysis of RCTs is that comparing meat greater than one serving per day versus less than one serving per day had little effect on cardiovascular disease risk factors, including cholesterol levels and blood pressure. So these are all listed here. The mean difference, the 95% confidence interval includes the mean difference, so none of these are significant. And the number is the number of studies. So there were almost 40 studies for cholesterol, but only 6 studies included for systolic and diastolic blood pressure. The authors conclude that the results from this systematically searched meta-analysis of RCTs support the idea that the consumption of more than a half a serving of total red meat per day does not influence blood lipids and lipoproteins or blood pressures. Now, this is a study published in 2019, and this was a similar study, although in addition to comparing all different diet comparators, these authors teased out different diet compositions into 7 different types of dietary patterns, ranging from high quality plant proteins, poultry and all types of fish, fish only, poultry or chicken, animal proteins, carbohydrates, and usual diet. So when they found, like the previous study, no significant differences between red meat and all dietary comparators. So when they lumped everything together, they didn't see any changes in blood concentrations of LDL or HDL cholesterol or blood pressure. They did find that triglycerides were higher for red meat compared to all diet compositions lumped together. It was higher. The mean weighted difference was 0.065 millimoles per liter. Confidence interval actually was 0 to 1.29. So not, so just on the borderline of significance. Now, when they looked further and started to look at these specific diet comparators, they found that substituting red meat with high quality plant protein sources led to more favorable changes in blood lipids and lipoproteins. They also found that substituting red meat with fish or low quality carbohydrates did not lead to more favorable changes. So again, it really becomes important to consider the composition of the comparison that you're making. And if you're lumping everything together or you're making comparisons to specific diets. Now, it's sort of interesting that red meat and fish, there were no significant changes. And probably that has to do with the type of red meat that was being consumed. The author suggests that if it were lean red meat with fish, might have contributed to that finding. The other thing is, as you get into these more specific comparisons of diets, you have smaller sample sizes. So those are a couple of RCTs. There are a lot of cohort studies that have been done. We certainly don't have time to go through those today, but let me just present recently published in 2021. This was an umbrella review, an overview of systematic reviews looking at unprocessed and processed meat. So a little bit different than the RCTs we were just looking at. This really did focus on processing and the association with cardiovascular disease. So these authors did their literature search. They found 3 systematic reviews that met their criteria. They found that each of these, using AmStar 2 rating, were rated as critically low. On the top half of the table is the meta-analysis of associations between intake of unprocessed red meat and CVD and stroke. And what they found was that the meta-evidence was graded as moderate for intake of unprocessed red meat and association with higher risk of stroke. They don't see what they concluded is that the evidence was very low for CVD and couldn't make any conclusions. But for stroke and unprocessed meat, there was some moderate evidence using the Nutrigrade rating for an association with unprocessed red meat intake and stroke. The bottom half of the table is processed meat and looking at risk of CVD, CHD, and stroke. And based on their Nutrigrade grading of the evidence, their meta-evidence was graded as moderate for intake of processed meat, which was associated with higher risk of CHD and stroke. They couldn't make any conclusions about CVD in general because the evidence was very low. So that's a little bit of just a touch of the literature on the cohort studies and RCTs looking at relationships between meat intake, processed meat, and further processed meat, unprocessed meat, and what we see with a lot of these studies is that there's problems with defining and classified meat. And that is why I wanted to talk about definitions first, because this is something that's plagued this literature for probably since the 1950s, and how people defined it. I saw one paper where they just classified all pork was processed, making the assumption that pork is mostly processed. So they just lumped all pork consumption in with processed meat. I'm not sure that, I think that clouds the results a little. Observational studies, usually there's inadequate adjustment for confounders, lack of repeated measurement of diet, and some of the FFQs are not validated. And for RCTs, that lack of validation of dietary assessment tools also is a problem. For a lot of studies, the tool might be validated for nutrients, but not foods, but it's used to gather information on the foods anyways. For RCTs, there's a lot of missing data, and diets are generally not controlled. There's been a few RCTs with controlled diets, but generally not controlled diets. With all these sorts of issues and knowledge gaps, we proposed to conduct an RCT, clearly defining what we were going to feed, with a highly controlled diet on different dietary patterns and mostly focused on CVD risk factors. Talk a little bit more about some of our outcomes later on. So our hypotheses, these were three of our hypotheses. LDL cholesterol and other blood lipids and lipoproteins will not be affected by meat processing, but will be affected by the quality of the background diet. 2, consumption of processed compared to unprocessed meat will increase peripheral blood pressure in an average quality diet, but not a high quality diet. And 3, when consuming processed meat, a higher quality diet will result in improved outcomes compared to an average quality diet. So you can see a lot of our questions are, what happens if we're eating a higher quality diet? Does that improve some of the potential negative outcomes of consuming processed versus unprocessed meat? So let me just go through some of the details of the study, the design, menus, our subjects, and our statistics. So this was a 2x2 factorial design. We had 2 levels of meat processing, our minimally processed meat and our further processed meat. We also had 2 levels of diet quality of the background diet, an average HEI diet and a high HEI diet. And I know many of you are familiar with HEI. This is a measure of diet quality used to assess how well a set of foods aligns with key recommendations and dietary patterns published in the DGAs. So 4 different diets where we overlaid meat processing and diet quality. Now, I know I've been talking a lot about minimally processed and further processed, and believe that is the best way to sort of describe what the meat is. Having said that, going forward, I'm going to talk about unprocessed and processed meat, just to keep it a little bit easier for us to think about. Just a little bit more about HEI. Again, I know many of you are familiar with this. This is a way to score a diet from 0, a dietary pattern from 0 to 100, based on the inclusion of certain amounts of different foods and restricting certain foods, shown on the bottom. And we decided that we would use HEI scoring to help develop our diets. So what foods did we use? For our processed diets, we used hot dog, corned beef, beef jerky, and bologna as beef items. And in the unprocessed, and didn't matter if it was higher or average HEI, the unprocessed, we used roast beef and ground, 90-10 ground beef. We used bacon, bratwurst, breakfast sausage, and bologna. This was a combination bologna for our processed menus, and pork chop for our unprocessed menus. And for poultry, we used deli chicken breast, deli turkey breast, smoked turkey sausage, and then we used roast chicken breast and roast turkey breast as our unprocessed. So we're trying to swap out meat for meat. If we had beef, say hot dog in the unprocessed diet, we would use roast or ground beef in the unprocessed, to try and keep everything as similar as possible. This is the composition of the diet. This is actually analytical work from samples that we collected. If we first look at protein and fat, the unprocessed diets were a little bit higher in fat as a percent of energy and a little bit lower in protein. And this is probably because those processed meat items generally have a little bit higher fat and less protein in them. But that quality of fat, it's usually the same sort of fat, whether you have beef roast or beef hot dogs. The hot dogs are going to have more fat, but it's going to be the same quality of fat, and that's going to be important. If we look at carbohydrate, our diets were, the average HEI diets were higher in carbohydrate and lower in dietary fiber. Again, this is by design. This is how we needed to get a high HEI diet. We had to get more fiber in the diet, and we had to use fewer refined grains and more whole grains and legumes and things like that. If we just take a look at the sodium and potassium, as you might expect with the processed meat, we had higher levels of sodium, 3909, compared to 2992 in the two average HEI diets, and also higher sodium in the high HEI processed menu versus the high HEI unprocessed menu. So our lowest sodium, as you might expect, was the diet that was unprocessed meat with high HEI. And again, this is by design. It's part of the dietary guidelines for American to limit salt. So we were able to achieve that. Sodium was highest was with the high HEI diets, higher with the high HEI diets, and lower with the lower average HEI diets. Just to put the HEI scores in context a little, our average diets were 50-55 score of HEI, and our high HEI diets were 86 and 94. To put that in context, in Haines, males' average HEI score was 56 in the last reported cycle and 60 for females. So we're in that ballpark, 50 to 55, 56 to 60. And we were able, through our diet development, to get high, very high HEI diets. Oops. So again, we looked at a little bit about the types of beef, pork, and poultry we used. These are the amounts. So a total of about 3 to 4 ounce equivalents per day of beef, pork, and poultry. We did have some other protein sources in these menus, fish and legumes, again, to achieve our HEI scores. But this was a study mostly of red meat. Most of the meat was from beef and pork. But we did include some poultry because we wanted to include some processed poultry, because deli smoked turkey breast and chicken and things like that are very popular, and we wanted to include them, and we wanted to swap out the processed poultry for poultry. Just to give you an idea, again, from Haines, what is typical intake that's reported in Haines, 50th percentile is about 0.8 and 2.3 ounce equivalents per day at the 95th percentile. So we were definitely at high intakes, especially on the processed side of things. We assessed for eligibility 120 people. We randomized 60. We allocated 62 different sequences. Talk about that in a couple of minutes. We lost the follow-up. We had 3 people who dropped before we collected any data. Then we had some dropouts during the different dietary intervention periods. We had 43 people complete all 4 diets and partial data that were included in our statistics from 14 people. These are the characteristics of the subjects that we randomized: 26 males, 34 females, mean age of 53, mean body mass index of 27, and cholesterol levels and blood pressures, as you would expect from a group of 53-year-olds. 15 of these people were taking antihypertensives for at least 6 months. We include people taking antihypertensives in our studies if they've been on a stable dose for 6 months. The intervention, again, as I said, this was a highly controlled dietary intervention. All food was weighed to the nearest gram. Subjects were fed at weight maintenance. They would come into our facility Monday, Wednesday, Friday. We would weigh them. We would make adjustments to calories to maintain weight. We would interact with the subjects, go over paperwork and things like that. And they would take their food off site and they would consume it off site. After randomization, we collected 1 fecal sample, 2 blood samples, and we collected blood pressure on 2 separate days. And at the end of each of the dietary intervention periods, which each lasted 4 weeks, we again collected a fecal sample, 2 blood samples on 2 consecutive days, and blood pressures on 2 consecutive days. And for the blood pressure, at each time point, we collected 3 measures. There was a week break in between each of the dietary intervention periods to help with compliance. The study was powered to detect a 10% change in LDL cholesterol. This was 4 treatments, complete crossover. Everybody consumed each of the 4 diets. With 4 treatments, there's 24 possible sequences. We selected 12 of those 24 so we could get replication on sequence. And subjects were randomized to a sequence based on sex, age, body mass index, LDL cholesterol, and systolic blood pressure, those that we collected at our screening. The analysis was done as a general linear mixed model. We used baseline value of the outcome as our covariate along with age, sex, BMI, and calorie intake. We used sequence and sequence by treatment interaction to assess a carryover effect. And we have not seen any carryover effect in any of the data that we're going to look at today. So there was no carryover. We have made adjustments because of the number of outcomes that we plan to have in the complete study. And then also within an outcome, we made a Bonferroni adjustment when we're looking at pairwise comparisons. So let's look at some of the data. These are some, again, preliminary data. These data will be adjusted once we get final statistics run and we know how many total outcomes there were. So 2x2 factorial, we can look at the diet effect across meats and the meat effect across diets. If there's an interaction, then we have to look at each of those separately. With triglycerides, there is no interaction between HEI level and meat. So in the bright red bar is the average HEI diet with processed meat. The dark red bar is average HEI with unprocessed meat. The dark green bar is high HEI with processed meat. And the bright green bar is high HEI with unprocessed. You'll see the same sort of graph in the next few slides. So there is no effect of the type of meat on circulating level of triglycerides. There is an effect of HEI on circulating concentration of triglycerides. The average HEI diet results in higher triglycerides, about 11% higher than the high HEI. So the 2 red bars are significantly higher than the 2 green bars. Higher triglycerides after consuming the average HEI diet compared to the high HEI diet, no effect on meat. This black bar is that one hypothesis that if you're eating processed meat on a high HEI diet, that will overcome some negative effects of consuming processed meat on an average HEI diet. That is significant. That is highly significant. But again, this is probably an HEI effect. But if you're eating processed meat with a high HEI diet, there is no difference compared to eating unprocessed meat with a high HEI diet. So in this case, high HEI diet does lower triglycerides compared to an average HEI diet when you're consuming processed meats. Let's talk a little bit about cholesterol. LDL cholesterol, we're interested in 2 things: the amount of cholesterol in the LDL particle and also the size of the LDL particle. So LDL, low density, lipoproteins are defined by their density, shown in the blue. And LDL, based on its chemical composition, what lipoprotein there is and what density it is, falls in a range of densities and diameters. So you can have larger LDL particles and smaller LDL particles. the smaller LDL particles are associated with higher risk for coronary heart disease, whereas larger HDL particles are associated with lower risk. So for the same amount of cholesterol, depending on how that's packaged, you can still be at higher risk or lower risk. And this is something that isn't routinely done, but is clinically relevant, and something that we routinely do in our studies is look at the distribution of sizes of LDL particles. Let's first look at LDL cholesterol concentration. Just like with triglycerides, we don't see any interaction between HEI and meat, but we do see an HEI effect. We don't see an effect of meat. Average HEI diets results in higher LDL cholesterol, about 9% higher than the high HEI diets. So again, the two red bars are higher than the two green bars. There is no effect of meat processing on LDL cholesterol. When consuming processed meats, a higher HEI diet does improve LDL cholesterol, but again, that's probably being driven by the HEI and not the meat in and of its, by the HEI. The effect of HEI and LDL cholesterol is possibly being driven by higher saturated fat intakes. So let's look at that. These are the diets. We've looked at the total fat in the diet, percent of energy. This table shows the monopufa and the fascinic acid, the trans fatty acid that's naturally occurring. And if we focus on the saturated fats, what we see is that the average HEI diets are higher, 14 and 13% of energy, compared to the high HEI diets, which are about 9% of energy. And again, this is to be expected. This is being driven by how we design those high HEI diets, which in order to be a high HEI diet, you have to limit saturated fat. There are some small differences in percent of energy from saturated fat between the unprocessed and the processed. We see that here too, the unprocessed and the processed, relatively small changes, but the big changes are are driven by the HEI content of the diet. And we know, one thing we know about LDL cholesterol is that saturated fats increase LDL cholesterol. So I think that this difference we're seeing is mostly being driven by the saturated fat content. Now, we talked a little bit about particle size. We actually don't see any effect of either HEI or meat processing type on LDL particle size. And this is the small particle. This is what we generally focus on. What about HDL, good cholesterol? Again, no effect of meat on HDL cholesterol. There is an effect of diet. Average HEI diets result in higher concentrations of HDL cholesterol. So the lower quality diet, higher good cholesterol. It's about 10% higher than the high HEI diet. There is no effect of meat processing on HDL cholesterol. And again, looking at the black, when consuming processed meat, a higher HEI diet results in a lower HDL cholesterol. And again, this is an effect probably being driven by the saturated fats. We know that saturated fats increase LDL cholesterol. Saturated fats also increase HDL cholesterol. One thing that we like to look at are the ratios of LDL and HDL, or total cholesterol to HDL cholesterol, because this incorporates a perspective of both the changes in LDL and HDL cholesterol. And some people think that the ratios are more predictive of risk than LDL or HDL independently. When we look at ratios, we don't see any effect of diet, of background diet, HEI, and no effect of cholesterol. And this is because those magnitude of changes for both LDL and HDL are similar. So as HDL is going up or down, LDL is going up or down as well. and the net result is no change in the ratios. Let's talk a little bit about blood pressure. We'll start with systolic blood pressure. We're presenting these data a little bit differently than the previous data we've looked at because there's a significant interaction between treatment and baseline systolic blood pressure. So when we use our baseline value as our covariate, we look at interactions with treatment. If there is an interaction with treatment, then we have to look at the data a little bit differently. Subjects with a baseline systolic blood pressure at a higher level, at our 70th quantile, which was 123 millimeters of mercury and above, so people in this range showed an evidence of a dietary effect driven by meat processing. Processed meats in these people with higher blood pressures yield greater systolic blood pressure than unprocessed meats, regardless of the HEI of the diet. This was significant at the 0.001 level, and it was about 3 to 5 millimeters of mercury, 3 millimeters here and 5 millimeters here. When consuming processed meat, a higher HEI diet does not improve systolic blood pressure. So these lines are not significantly different. It's about a 3 millimeter difference, but it's not significant. Probably it's effect of meat processing. This effect that we're seeing of meat processing on systolic blood pressures possibly being driven by higher sodium and lower potassium intakes. If we look at diastolic blood pressure, we don't see that same interaction with baseline, so we can look at these data like we've looked at some of the cholesterol levels. There is no effect of dietary pattern, HEI score on diastolic blood pressure, but there is an effect of meat processing. Processed meat increases diastolic blood pressure about 2 millimeters of mercury, and this is statistically significant. When consuming processed meat, a higher HEI diet does not improve diastolic blood pressure. These are not statistically significant. And again, the effect of meat processing on diastolic as systolic as well, probably being driven by sodium potassium. So just to remind you, our highest sodium is on the processed meat diets, 3909, 3301, lower sodium on the unprocessed meat diets. Again, probably because those processed meats are contributing extra sodium to the diet, additional sodium to the diet. And potassium levels, the highest potassium level we have is on the high HEI diet unprocessed. So we think that's the reason for the results that we're seeing. So just as we begin to wrap up, some caveats about what we did, some strengths and limitations. This was 4 weeks of feeding for each treatment period. That's enough time to see changes in blood pressures and cholesterol levels. Meat intake was 3 to 4 ounce equivalents per day. So that is typical for total meat, but certainly for processed meat, we were pushing the envelope. But we wanted, again, to keep everything consistent and feed meat at a typical level and then swap out. This was a highly controlled diet. We believe we were able to achieve our dietary goals where we were able to separate HEI scores of the diets. We used HEI to qualify diet quality, quantify diet quality. There are other ways to do that. We have chemically analyzed the diet, so we know what we were feeding, and the way we did our collections work was quite rigorous. So let's go back to our hypotheses. LDL cholesterol and other blood lipids and lipoproteins will not be affected by processing of meat, but will be affected by the quality of the background diet. Now, we set forth this hypothesis because, again, the thing that really is going to drive LDL and HDL is the quality of the fat, in particular, how much saturated fat. And as I mentioned earlier, even though a roast or ground meat, 90-10, 80-20, is going to have different amounts of fat than in something that's minimally processed compared to further processed. The quality of the fat will be similar. And we found that this was mostly true. Consumption of processed compared to unprocessed meat will increase peripheral blood pressure in a lower quality diet, but not a higher quality diet. Blood pressures were higher regardless of the HEI score. When consuming processed meat, a higher HEI diet will result in improved outcomes compared to an average HEI diet. This was generally true for triglycerides and LDL, not true for HDL cholesterol, again, being driven by saturated fats, but also true for our ratios. And there was no difference in blood pressure. So, a couple of summaries and conclusions. What meat to eat? Well, this was, we believe, the first controlled diet intervention of unprocessed and processed meat. And again, this really focused on red meat versus processed meat. Red meat being, in this case, a combination of beef and pork. The results thus far suggest that processed compared to unprocessed meat, 1, increases blood pressure 3 to 5 millimeters of mercury. This is consistent with epidemiological data on stroke that we looked at earlier. But the effect on systolic blood pressure in our study was only seen in people who had higher systolic blood pressure to start with. The results thus far suggest that processed compared to unprocessed meat does not affect blood lipids, including triglycerides, LDL, and HDL. And again, this is consistent with some of the meta-analyses that have been conducted. Results thus far suggest a higher HEI diet mostly improves blood lipids regardless of the type of meat consumed. So is it the meat or the diet? A couple of final thoughts. Talking about what we do is really important, and clearly defining what we're measuring is really important. And historically, there's been a lot of unclear terminology used, and this just gets propagated from individual studies into meta-analysis, into umbrella reviews, and it just creates a lot of confusion. And this is something that I think has plagued most of the research and has plagued some of the recommendations. In our study, diet quality impacts are more pronounced than the type of meat. Which also means that lumping together different types of meat in terms of dietary guidance, like saying all red meat and processed meat together, that's not really going to be as informative for making science-based recommendations. So let me stop. Let me just say we are working on some additional data. We're doing metatranscriptomics on the fecal bacteria, the fecal samples that we collected, and some other microbial metabolites. We've talked mostly about cardiovascular disease, and then there's that whole side of things on cancer, which is obviously harder for us to understand, especially in this type of a study. But we're hoping to maybe get some insights as we do metatranscriptomics and some other continuing analyses on the samples we collected. So I'll stop there and see if there's any questions.
Well, thank you, David, for a very interesting and insightful presentation and some wonderful research. We do have a couple of questions. First question comes from Larissa Constantino, and she asks, are there any differences in their impact on health between organic and regular meat? Some people may be concerned with the agrochemical residues, antibiotics, hormones, etc., used in animal husbandry in meat. Is this really a concern?
Well, good question. Big question. Obviously not something that we incorporated into our study design. Is it a concern? I'm not sure yet. I don't know if we have enough data to draw conclusions. Certainly, there are options in the marketplace. So if people have concerns or might be at risk for something, that there's choices that people can make. That's a huge question, and whether it's meat or produce, there's pros and cons. There's pros and cons to all of these decisions, and usually they're complicated sorts of nuanced data that have to be considered.
The next question comes from Alice Lichtenstein, and she thanks you for a very interesting talk and states that the triglyceride findings are intriguing. Triglyceride concentrations usually track with relative proportion of carb and fat, assuming protein remains constant. Do you have any thoughts on this issue relative to your data?
I have to go back and look, because we also did have protein changing a little, because as we swapped out replacement on a calorie for calorie basis for the processed and unprocessed meats, we were impacting protein. I think our lowest protein was about 70% of energy on one of the diets and 22% of energy on the other. So Alice, I'm not sure if that's a big enough difference in protein to sort of complicate the fat carbohydrate impact. Something to look into, and I'd be happy to talk with you further about it and get your thoughts.
The next question comes from Jiandong Wang, and he says, you mentioned that there was no carryover effect in your study. What biomarkers did you use specific for processed meat?
Well, when I talked about carryover effect, that was looking at the statistical analysis. So with a crossover study, we're always concerned about a carryover. The impact of a previous dietary treatment will have on the subsequent dietary treatments. So we look at that, because if there is a carryover effect, we actually can't further analyze all the data. So we used both the sequence and the sequence by treatment interactions from a statistical perspective to look for carryover effect. We don't have biochemical biomarkers, if that's the question, to look at, are we seeing something in the blood from a previous dietary treatment? That's not how we look at carryover. It's more from a statistical perspective.
Okay, and next question comes in from Barbara Lyle, and she asks, in comparing the high versus average HEI diets, can you summarize what was the most important differentiator? For example, is it the primary difference in total fruit and vegetable intake, or was it in the type of oil used, or what?
Barbara, with these sorts of dietary patterns, we really can't differentiate. We don't know what might be having the biggest impact. We in designing the diets, we did everything we possibly could to get as high an HEI score as possible. So that included fruits, vegetables, limiting saturated fats, limiting simple carbohydrates, and trying to incorporate as much seafood and everything else that we had to incorporate to get as many points as we possibly could. And if you remember that a processed high HEI processed diet, the HEI score was 84, and again, that's the best we could do. We would have liked to get it over 90, but again, by having processed meat in there and more saturated fat and more sodium, we were penalized on our HEI scores for that. So again, it's the dietary pattern, and that I'll say our original study design did not include the two dietary patterns, and that obviously doubled the size of the study, doubled the trouble. But I'm really grateful we did that, because I think it really pointed out the critical importance of dietary pattern. It really shows that you can eat things like processed meat if you're eating a better diet, if you're eating a healthier diet, as we like to describe it, at least by HEI score.
Okay, well, seeing no more questions in the queue, I want to again thank you for a very interesting presentation. And
Thank you.
okay, and I think with that,